Hello everyone, welcome to Decode Science. In this video, we are going to discuss about a very important technique in molecular biology, that is the CRISPR-Cas mechanism. CRISPR-Cas mechanism is generally referred to as the immunity mechanism in the prokaryotic cells, as it is helpful for the as it is helpful for the prokaryotic cells to fight against their pathogens. So let's start our discussion with this technique. So first point is the introduction. So CRISPR is a family of DNA sequences in the genome of prokaryotes and archaea. And CRISPR simply stands for the clustered regularly interspaced short palindromic repeats, and Cas stands for the CRISPR associated protein. So CRISPR is nothing but the family of DNA sequences. For example, like CRISPR is specifically a DNA sequence which is generally present in the prokaryotes and archaea, and which is helpful for the prokaryotic cells to fight against the different types of pathogenic infections, such as the bacteriophages infect the bacterial cells. So bacteriophages are the pathogens for the bacterial cells. So the CRISPR mechanism will be helpful for the bacterial cells to fight against the bacteriophage infection. So as I told you, CRISPR is nothing but a DNA sequence. So these are nothing but the palindromic repeats. Okay, the CRISPR is nothing but a small palindromic repeats which are interspaced. Okay, which are interspaced by a certain types of DNA sequences which are derived from the pathogens of that prokaryotic cell. If we'll take the example of the bacteriophage, so the palindromic repeats will be interspaced by the certain types of DNA sequences which are acquired from the bacteriophages in the bacterial cell. Okay, and Cas are nothing but the specific for the special types of proteins which are involved in the CRISPR-Cas mechanism. So in nature, CRISPR-Cas system is prokaryotic adaptive immunity mechanism used to clip the invading nucleic acid. So generally the CRISPR-Cas system is referred as the immunity mechanism for the prokaryotic cells because it is helpful to fight against their pathogenic infection. For example, in our body, we have an immunity mechanism which is helpful for ourselves to fight against the different types of pathogenic infections. So, similar way, CRISPR-Cas mechanism is also helpful for the prokaryotic cells to fight against this type of pathogenic infections. That's why they're called as the adaptive immunity mechanism of the prokaryotic cells. These CRISPR sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryotes. So as I told you that CRISPR is a DNA sequence which includes the palindromic repeats, small palindromic repeats. So these palindromic repeats are interspaced by a certain type of DNA sequence. Okay, these CRISPR sequences are derived from the DNA fragments of bacteriophages. So as these palindromic repeats are interspaced by a specific type of DNA sequence, that specific DNA sequences are generally derived from the bacteriophages. Bacteriophages are the viruses which infects bacteria. That means the bacteriophages are the pathogens of the bacterial cell. So bacteria acquire certain DNA sequences from their pathogens, that is the bacteriophages, which is getting integrated into their DNA, and that DNA sequence is referred to as the CRISPR sequence. Okay, these sequences used to detect and destroy DNA from subsequent infections. So as I told you that CRISPR-Cas mechanism is helpful to fight against the bacteriophage infection and all. So that's why the sequences which are derived from the bacteriophage infection in the CRISPR sequences are helpful to detect and destroy the DNA from subsequent infection. So if a bacteriophage will infect that bacteria again in future, so this bacteriophage derived DNA sequences will be helpful for the bacteria to destroy the DNA of that bacteriophage. Let me tell you more clearly with the help of diagram. Okay, so here you can see this is the bacterial cell, and here this is the bacteriophage which has infected this bacterial cell. And as we all know that the bacteriophage insert their genetic material into the bacterial cell, and then with the help of the host replicating machinery, this bacteriophage DNA will be get replicated, and then it will form the entire virus structure, and then it will burst the bacterial cell and will come out. Okay, this is how this cycle works. So here you can see the genetic material of the virus has been integrated into the. So here you can see the genetic material of the virus has been integrated into the bacterial cell. Now what will happen is if somehow this bacteria will survive. So what the bacteria will do? Bacteria will cut this bacteriophage DNA and take out a specific DNA segment from this entire bacteriophage DNA and will integrate that specific DNA part or the DNA segment within the bacteria's own DNA. Okay, so this the black part. Okay, this black part are nothing but the bacteria's own DNA, and these are nothing but those small palindromic repeats. As I told you that CRISPR sequences include the small palindromic repeats. This is the one repeat. This is the another one. So bacteria will cut this bacteriophage DNA and will take out a small DNA fragment or segment and will integrate that DNA segment within its own DNA, and this entire structure will be called as a CRISPR sequence. Okay, so this black part is the bacteria's own DNA sequence, which are nothing but the palindromic repeats, and this is the red, green, purple, all these are nothing but the DNA sequences which the bacteria has acquired from the different bacteriophages. So this is how the CRISPR sequence generally looks like. Next step is the CRISPR RNA formation. So as I told you that this is a DNA sequence. So now this DNA sequence will be get transcribed into RNA, and it will form a specific type of RNA which we call as a CRISPR RNA, and this CRISPR RNA will be helpful to fight or the detect the bacteriophage DNA and will destroy it. So what happens is as the bacteriophage has infected the bacterial cell and bacteria has integrated a specific DNA segment of that bacteriophage DNA into its own DNA and has formed the CRISPR RNA. Okay, so if in future, if this bacteriophage, okay, the same bacteriophage will reinfect the bacteria, okay, so what happens is this CRISPR RNA will also have a DNA sequence which will be similar to the not DNA sequence, sorry, it will have a sequence which would be complementary to the DNA sequence of that bacteriophage. Okay, because this DNA segment has derived from this bacteriophage itself. So that's why it will show the complementarity with the DNA of that bacteriophage, and it will just go and bind to that bacteriophage DNA, and with the help of certain types of proteins, the bacteria will destroy this bacteriophage DNA. And as the DNA of the bacteriophage will be get destroyed, so it won't be able to replicate and won't be able to survive. So this is the normal overview of the CRISPR-Cas mechanism, that how it generally works. Okay, now let's talk about it in detail. So just to understand the CRISPR-Cas mechanism, first we need to understand this important thing. First one is the CRISPR locus, and second one is the mechanism. The mechanism is again subdivided into three steps. That is the spacer acquisition, processing of CRISPR RNA, and the interference. Okay, so let's talk about it. So let's discuss them one by one. The first one is the CRISPR locus. So this is the diagram of the CRISPR locus, and this is the description. So first of all, as I told you that the CRISPR is nothing but a DNA sequence. So let me tell you that this is the DNA sequence of the bacteria. This blue part is the bacteria's own DNA, and after the infection of the bacteriophages, the bacteria will cut the bacteriophage DNA and will integrate a specific DNA fragment into its own DNA, and because of that these two palindromic repeats will be get interspaced with the help of this DNA fragment, which we call as a spacer sequence. Okay, so here we have mentioned is that blue part is the repeat sequence and the red part is the spacer sequence. So spacer sequence is the DNA sequence derived from the bacteriophage DNA. Okay, here we have the Cas genes. These Cas genes are very helpful in this process. These Cas genes are important for encoding the certain types of Cas proteins which are helpful for the spacer integration or the acquisition process that we'll see further. So this is the leader sequence where generally the promoter region and all lie. So this is the structure of the CRISPR locus. For example, like so this is the structure of the CRISPR locus. So here you can see the CRISPR locus consists of the repeated sequence, each approximately 30 nucleotides long and highly conserved within the given cluster. So this is the blue part, which is the repeat sequence, is generally 30 nucleotides long, okay, and which is separated by a unique spacer sequence of similar length but highly divergent. So here you need to know this, that the spacer sequence which the DNA, bacteria derived from the bacteriophage or acquired from the bacteriophage is of the similar length. For example, like this is the 1, 2 and the n number of spacer sequences are acquired. So if the bacteria will acquire the spacer sequence from the bacteriophage number 1 and it will acquire the DNA sequence from the bacteriophage number 2. The length of this both these spacer sequences will be same, that is near around 20 nucleotides, but they will be different from each other, okay, as the DNA sequence of the two different bacteriophages will be different. So the sequence will be different, but the length of the two spacer sequences will be the same. And at one end of the array, a leader sequence is present, often 80 rich. As I told you, this is the leader sequence which contains the promoter region. That's why it is 80 rich sequence. And a set of conserved protein coding genes, that is Cas gene, is present. So Cas genes are helpful for the encoding the Cas proteins, which are important in the CRISPR mechanism. Now let's talk about the mechanism. Action of the CRISPR-Cas is usually divided into three stages. First one is the spacer acquisition or the adaptation. Second is the processing of the CRISPR RNA and maturation. And third is the DNA or RNA interference. So these are the three main important steps of the CRISPR-Cas mechanism. So first step is the adaptation process or the spacer acquisition process. So the first step is to capture a phage DNA and insert into a CRISPR locus in the form of spacer. So, and two proteins, Cas1 and Cas2, are responsible for the insertion of the spacers into the CRISPR Cas. So, as I told you here, okay. So, when the bacteriophage infects the bacteria, so what happens is bacteria cut a specific DNA segment of the bacteriophage DNA and will integrate that DNA segment into its own DNA. So, this entire process is called as the spacer acquisition or the adaptation process. And as I told you that Cas1 and Cas2 proteins are responsible for this process. Okay. So Cas1 and Cas2 protein, these two proteins form a complex that is required for this adaptation process, and the integrase activity of Cas1 is required for spacer acquisition, whereas Cas2 show the endonuclease activity. So Cas1 and Cas2 protein, they generally have the integrase and the endonuclease activity. So they will cut the bacteriophage DNA and will integrate the DNA segment of that bacteriophage DNA into the bacteria's own DNA. Okay, so the spacer acquisition or the adaptation process is generally carried out by the Cas1 and Cas2 proteins. Okay, so new spacer is inserted proximal to the leader sequence, and the sequence in the virus that will become a new spacer is called the protospacer and is found close to a PAM or the protospacer adjacent motif sequence. So the sequence in the virus, for example, like this is the bacteriophage DNA, and if the Cas proteins, Cas1 and Cas2, will cut the bacteriophage DNA like this, okay, and this is the DNA segment, this particular DNA segment of the bacteriophage is going to be inserted into the bacterial DNA. So this DNA segment is generally called as the protospacer region. Okay, so this DNA segment will be get inserted into the bacterial DNA like this. So this is the bacterial DNA. And this is the bacteriophage DNA segment. Okay, so this particular segment will be get inserted into the bacterial DNA. So this segment which has been get inserted into the bacterial DNA is called as a protospacer. Okay, and is found close to a PAM region. So for example, like this region, okay, this region which is adjacent to this protospacer region is generally called as a protospacer adjacent motif for the PAM sequence, which is very very important in the interference step. That is the the destruction of the bacteriophage DNA when takes place. So that for that process, this PAM region is very very important. So I hope you will understand till now.